Hello everyone, I'm Noémie Hévin and I'm going to present a study on the diversification of the weevil genus Phlyctinus endemic to the Cape Floristic region. The Cape Floristic region in South Africa is a well-recognized biodiversity hotspot hosting a high diversity of endemic plants. By contrast, insect diversity has long been considered poor, but recent studies revealed higher than expected level of arthropod diversity, especially in phytophagous insects. We can therefore ask ourselves if the diversity of phytophagous insects is as poor as first considered, and if not, what is their diversification process? Is it mostly driven by abiotic factors, as for the plants, or by the plant diversity itself? Weevils are often used as models to investigate diversification patterns in relation to biotic and abiotic drivers, thanks to their high diversity, worldwide distribution, and highly diverse morphology and biology. Here we focus on the genus Phlyctinus, which is apterous and endemic to the Cape Floristic region. This genus was originally thought to be monotypic, but in 2020, Aron et al. Described 5 other species and postulated that this species complex may consist of even more species. Among the known species, Phlyctinus callosus and Xerophilus are major pest species on apple and vineyards, causing this kind of damages. The full picture of the diversity of this genus is far from being complete, since preliminary investigations on the morphology and molecular diversity of recognized species have revealed potential additional cryptic lineages. The first aim of the study was therefore to investigate the evolutionary relationships of the genus Phlyctinus. Phlyctinus, as I said, are apterous and endemic of the Cape Floristic region. They are found in humid habitats, mainly on a few genera of Asteraceae, thus suggesting the potential role of host plants in their diversification. In this context, exploring the diversity of the genus Phlyctinus can provide novel insights on the origin of the Cape Floristic region phytophagous insect fauna. The second aim of the study was therefore to assess the speciation processes that have led to the present diversity of the genus. To do that, we considered 2 main speciation patterns: allopatric, by looking at geographical occurrences in light of late Cenozoic environmental changes, and sympatric, by looking at host plant associations. To do that, thousands of Phlyctinus were collected and 170 were sequenced for 2 mitochondrial and 4 nuclear genes using both Illumina and Sanger sequencing. To infer the evolutionary relationships between the species, phylogenetic analyses were made using maximum likelihood and 2 branch support estimators. Then, 21 species delimitation analyses were made on concatenated dataset or fragment genes using either multilocus coalescent-based, distance-based, tree-based, and parsimony-based methods. Then, a match ratio was used to see the congruence between a priori defined lineages and putative species inferred by the analysis. Finally, dating analyses were conducted using 4 secondary calibration strategies based on substitution rates. So first, we provide new insights on the evolutionary relationships of the genus Phlyctinus. The phylogenetic tree from maximum likelihood analysis is represented here. It confirms the genus Phlyctinus as monophyletic and well supported. The lineages selected for the study are highlighted by colored boxes. On the right, results of the species delimitation analysis are represented by vertical bars colored in green when the putative species inferred by analysis correspond to selected lineages, in yellow when they are splitted, and in orange when they are lumped. Overall, the analysis yield a height range of putative species estimates, often under or over splitting species, which is highlighted by low match ratios. This is not unexpected, hence the importance of assessing species boundaries within an integrative framework. Therefore, considering molecular analysis, but also morphology, geographical range, and ecology, a total of 12 lineages were selected. First, the 6 previously described species are supported by clear morphological traits, by their monophyly, and by some of the molecular analysis. Second, 2 undescribed lineages are morphologically distinct, monophyletic, and well recognized by molecular analysis, and will be described in the near future. And lastly, the 4 remaining lineages are monophyletic but recovered by only a few of the molecular analysis. 2 of them are morphologically different from the other species, but not between them, and have overlapping distribution ranges on the same host plants, while the 2 others shows no clear morphological characters and are distributed in distinct days. They are selected for the study, but calls for additional investigation to be considered as new species. Second, here is what we found about the diversification of the genus Phlyctinus. Our results do not suggest a major effect of host plant associations on the diversification of Phlyctinus species. Indeed, in natural habitats, Phlyctinus species from distinct lineages are generally found on the same host plant in the same locality, whereas the inferred species pairs are not found in same patry. Instead, Phlyctinus species appear to be quite opportunistic. Such a pattern does not provide any support for ecological speciation in some patry. On the left, you can see the dated phylogeny of the genus as well as the sea level oscillations. The gray points represent raw data with many oscillations during Quaternary, and the blue line represents the overall trend. On the right, you can see the geographical distributions of the 3 main groups. The early diversification of Phlyctinus during the Miocene led to the emergence of 3 main groups currently restricted to specific regions. We postulate that the progressive aridification during the late Cenozoic strongly constrained the diversification of the genus by trapping populations in the most humid habitats of the Cape Floristic region, which are the coastlines and the inland valleys. Most of the diversification of Phlyctinus species occurred during the late Pliocene and the Pleistocene, highlighting contrasted diversity between inland and western coast groups with only 1 lineage compared to the southern coast group with 10 lineages. We hypothesize that this diversity pattern is best explained by the role played by the interplay between geographical barriers and sea level oscillations. In the western coast group, which consists of 1 lineage, we assume that the lack of elevation allows gene flow between populations, explaining the low diversity. In the inland group, which also consists of 1 lineage, there are many mountains, but Phlyctinus are found in connected valleys. We made 2 hypotheses to explain the low diversity. First, even if there are mountains, the valleys could remain connected, allowing gene flow. Second, aridification without sea level oscillations as climatic buffers could have resulted in extinction. In the southern group, which consists of 10 lineages, we assume that the high diversity could be the result of the interplay between the mountains separating bays and the sea level oscillations that may have acted as climatic buffers. It is consistent with the current observation of some Phlyctinus species of this group, with some inferred species being endemic of a single bay, as you can see on the little map. To conclude, the integrative approach followed in this study provides support for the existence of additional cryptic Phlyctinus species, echoing the idea that insect diversity in the Cape Floristic region is likely greatly underestimated. Host plant association patterns provide no support for sympatric speciation in relation to host use, and allopatric speciation, instead, appears to be the main process of speciation for Phlyctinus weevils. Overall, this study adds to the emerging corpus of research supporting the role of abiotic factors and allopatric speciation for generating arthropod species diversity in the Cape Floristic region. Thank you very much for your attention.